Good afternoon, everyone. First, I would like to thank the organizing committee for giving me this opportunity to talk about my research project. Actually, this is an update of my presentation, which I did last year. Just to remind you, we at my lab, we work with different viruses, but this is, I'm going to talk about use of quantitative comparative proteomics for study of molecular mechanism of host-virus interaction for PEDV and PRRS. For the purpose of this talk, what is important is just to remember that these viruses, they have a small volume of femtoliters of cytoplasmic content, and they are envelope viruses, so these envelopes, they are derived from the host membrane. So this is an important thing to keep in mind. Both viruses, like I said, envelope viruses, they are successfully modulating several cellular pathways. It was reported for both viruses that they can also induce microvesicles production. They modulate apoptosis, ER stress, signaling. They also modulate interferon response and can affect the cellular cell cycle. So I mentioned exosomes, microvesicles. Growing evidence actually show that indicate that they play an important role in viral pathogenesis. First, they can include viral proteins, express viral proteins. They can include, contain viral RNA, microRNA, and having all this charge, they are also the large microvesicles. They can also contain actually viral particles. So they are important for establishing productive infection and modulate cellular response. So after revising all this briefly literature regarding this host-virus interaction for these two viruses, I came with three hypotheses that at viral level, this dynamic should be a dynamic interaction between virus and cellular proteins, and therefore composition of viral particles should be cell type dependent. In other words, I believe that the same virus, genetically same virus produced by different cells can have different pathogenicity, infectivity, etc., etc. At cellular level, because viruses modulate intracellular levels, they use cellular machinery, so it could be expected that both viruses should up and down regulate several host proteins, and knowing this dynamics will help us to better understand this issue, the virus-host interaction, and help us to develop maybe or find new targets for the antiviral control. And finally, the exosomes, which are also part of this equation. So they're produced by infected cells, and there are already a couple of papers, at least few papers, showing that their proteomic patterns are different. So with this, we formulated some objectives and how to achieve or how to prove this hypothesis. Basically, what we propose is for first hypothesis, we propose to just produce viral particles from simian cells, which are usually used in labs, and also natural host cells, and analyze using a proteomic approach, quantitative proteomic approach, the exosomes and viral progeny. And also for studying the dynamics of viral host interaction inside the cells. So we just lyse the cells at different time points of post-infection and analyze the proteomic profiles. So very briefly, about how we did this work. As you can probably imagine, when you collect the super from the infected cells, this super have actually everything. It's a soup of everything. There are viral particles also, but there are microvesicles, cellular debris, etc., etc. So in order to be able to say with certain level of security what are the proteins which are encapsidated or associated with viral particles, we did so differential treatments, ultracentrifugation and treatments with some enzymes in order to get this highly purified or ultra pure, I will call them ultra pure viral preparation. So basically comparing proteomic composition of these three different preparation, I will be able to say what are the proteins which are associated with exosomes produced by infected cells, and which are the proteins which are associated only with viral particles. And we did a very vigorous control of quality. We measure using the PCR, we measure the content of viral RNA in each fraction of ultracentrifugation, protein content, and also titer, viral titer. Doing this, we know exactly which fractions are pure, they have a pure viral particles, and then when we do ultra-pure purification, treating this viral preparation with subtilisin. Subtilisin is an enzyme which will digest all basically microvesicles, and only viral particle will survive this treatment. So, and this is what we expected to see, like I said. When you have pure viral particles, you can say with certain level of confidence that these proteins, host proteins, are encapsulated in viral particle, and which are the host proteins which are associated with exosomes. So this is how it looks usually. You do a proteomics, proteomic digestion, and when you analyze the peptides, and you can see here for person PEDV, despite that they are morphologically and from the point of view also genetic structure, they are similar viruses, they behave differently in the same host. Basically, this is, I think it's Mark cells, and this is Vero cells. Both are from green monkey cells. And you can see that in semi-purified fraction, which has a lot of microvesicles, most of the host proteins are downregulated. And in case of PEDV, there are a lot of proteins which are upregulated. So, and this is actually the actual picture of this semi-purified fraction. I repeat, this is a fraction which has also microvesicles and viruses, and we compare with non-infected cells. So that is why we are sure that what we observe, downregulation or upregulation of host proteins, they are related to these microvesicles and viruses. And as you can see, certain functional groups like nucleic acid binding, signaling, metabolic process, cell cycle, and immune response proteins have been dysregulated by this by viral infection. That was in case of PRRS, and this is in case of PEDV. Basically, same group of proteins have been dysregulated. Additionally, for PEDV, we find that cytoskeleton organization proteins involved in the cytoskeleton organization have been highly dysregulated. Later, what we did, we also classified these proteins according to their subcellular localization, and you can see that for PRRS and PEDV, host proteins which are associated or localized in cytoplasm, nucleus, and plasma membrane have been upregulated or downregulated. What is interesting, like I said, in that semi-purified viral sample, which most of you use for your research, or maybe even you don't even do that semi-purification, but there are a lot of host proteins which are associated with microvesicles. In case of PEDV, they are really upregulated, while in case of PRRS, they are downregulated. So we were happy actually to confirm that the proteins, individual proteins which we find in our proteomics, profiles have been actually found by our groups. This is just a very short list showing that the hits, the targets like vimentin, heat shock protein 17, and applebags have been found by our groups in exosomal fractions. So then, like I said, we did gradual differential purifications, and this is a more pure viral preparation. And again, you can see that certain functional groups, proteins, have been affected. But what is interesting is absolutely different pie. As you can see, different proteins have been dysregulated in this fraction, which is now mainly viral particles, okay? So what I will expect, and this was actually proved by our data, that, for example, proteins associated with vesicles, as you can see, they are not really present. I should be honest with you. Biomarkers of exosomes like CD80 or CD86 and others have been still present in this pure viral preparation, but at very, very low levels, showing again like that our approach is correct. But at the same time, we saw upregulation and downregulation of abundance of certain host proteins, showing again that this approach is absolutely fantastic, and we have more, shorter list of proteins, but these proteins are encapsidated or associated with viral particles. And like I said, I really believe that these host proteins, they play important role in viral pathogenicity. They are not, some of them, of course, they are encapsulated randomly because they are just part of cytoplasm. But I'm pretty sure that few, maybe many of them are there because they should be there. They are specifically encapsulated. So we did this list, and just briefly for cellular part of the work, we did proteomic, like I said, study of changes along the viral infection. At different time points, we analyzed the proteomic profiles, and now, I can say, we're massaging this data, trying to find meaningful results. And for example, here on this cluster analysis, you can see that a group of proteins, and it is logic, which are close from a time point of view, they show similar patterns. For example, I don't see very well here, but it's 0 and 2 hours here. They're paired, as you can see. The only difference which we cannot still explain is that proteomic patterns at 12 hours and 48 hours of post-infection were more similar than the next, let's say, time point. In this graph, upset analysis graph, we try to show for each time point the group of proteins which have been downregulated or upregulated, and after doing this type of research, we come with this type of analysis, which actually confirm many of published data. For example, here you can see that type 1 interferon signaling pathway is downregulated, and you can see here another hit, which is cellular response to exogenous double-stranded RNA, this red one. So, and more importantly, we can now do correlation between the abundance of viral proteins, because at the same time, we measure abundance of viral proteins for each time point. And knowing the kinetics of the expression or abundance of viral proteins, now what we are doing is for each viral protein, we are trying to find association or correlation with certain individual host proteins or group of proteins. I just give you 2 examples, but I have already dozens of these graphs for each viral protein. In this case, you can see that we are showing 2 graphs. The graph for replicase, PEDV replicase, PP1A, and type 1 interferon signaling pathway proteins. And as you can see, there are certain correlations which you can now think about how to explain that. In this graph, we show expression or, yes, abundance of spike protein and proteins which are involved in entry to the cell. Basically, the taking home messages are different here. First, so viral particles, they are associated and they encapsulate certain host proteins which are important probably for a virus. If so, if it's true, and I believe it's true, then different subset of the host proteins encapsulated in viral particles produced in different cell types will change the pathogenicity and infectivity of viral particles. And doing all this global profiling, that is why I'm calling this project global profiling, because we study in host-virus interaction at different levels. Like I said, viral particles, exosomes, and cells. So we will be able to build all this network of virus-host interaction. We have now a lot of data and just need students who will work on this. So, and finally, I just want to thank my colleagues, members of my lab who participated in this project, my colleagues from Université de Montréal, Francis Baudry, Carl Carnion, Chantal Provost, and my colleagues from USA, Federico Zuckerman, Fernando Osorio, and Alexander Sakarchuk from Saskatchewan University, Canada. And thank you for your attention.